today when I dropped her off at school, and so she never complains at all. That's probably means that I'm not going to last the whole day. So, just waiting for that call. All right, here we go. Mitosis. That's what we're going to talk about today. We talked a little bit about it yesterday. It's one of the M phases that you can go through. The two M phases are mitosis and meiosis. Mitosis is the one that is going to produce daughter cells that are identical. Okay, so we can say that one parent cell produces two genetically identical daughter cells. Okay, we can say that this process is generally the process that your body cells are going to go through, and it's going to produce what's called somatic cells. So it produces somatic cells in animals. Okay, somatic cells are those cells that are non-sex cells that have the normal amount of chromosomes that the rest of the body has, and sex cells are going to have half as many. And meiosis is going to produce sex cells, but we'll talk about that next unit. Okay, so I want to talk really quickly about DNA organization. We talked briefly about it yesterday, but I wanted to touch on it again. So, when a cell is in G1 phase, okay, it is going to have one copy of its DNA. That's one DNA double helix, right? And that DNA is going to be pretty much spread out. There's going to be portions of it that are a little bit more tangled up, but for the most part, so here's maybe a tangled portion. Here's a more spread out portion, right? Here's a more tangled portion. Here's a more spread out portion. So you can see that for the most part, there's the ability for an enzyme like RNA polymerase to go in here and actually create RNA from the vast majority of this DNA double helix, right? And this is this form of DNA here is called chromatin. Okay. Now, if this cell gets the go-ahead to then start replicating, it's going to go into S phase, right? So if this is S phase here, what happens in S phase? Yeah, replication of the DNA. So we're going to have double the amount of DNA that we had before. So we've got our first strand here that's got the tangled up portion there. Second tangled up portion there. Okay, and then we've got another unique strand, right? We've got double the amount of DNA. So we've got now this strand, the same tangled up portion, another tangled up portion here, right? Hopefully you can tell there that that is twice as much, right? This is 2 times as much DNA. Okay, then it's going to go into the process of mitosis, okay? We said that a cell that is going through S phase and G2 phase, it's definitely going to travel on through. Can anybody think of what might happen to this cell if it had double the amount of DNA that it should have and it just stopped? What if it just stopped in G2 phase and it continued to live in G2 phase forever? What would that cell do that's different from another cell?
Just had double the DNA?
If it had double the amount of DNA.
It wouldn't just go into apoptosis.
It might go into, let's say it did not go into apoptosis. But let's say, because we talked about the cell cycle here, and we said that in G1 phase, that's really where the cell is the best at doing its job, and then it can go into G0 phase right around here if it really wants to, right? And that's when the cell is really good at doing its job. But what if it stopped right here before M phase, and it just like continued to live its life? What would it do?
The G0 phase.
Well, imagine it was like a G0 phase here, but how differently would that cell act? Because it had double the amount of DNA. Okay. Now, this is a really sort of conceptually important question, because this means, if you get this question, this means that you understand what cells are doing and why they're doing it, okay? Cells are stupid. They're not thinking about anything. The only reason a cell does anything is because there are spots on its chromosome, or spots on its chromatin rather, on its DNA that are open enough for mRNA to be made, and then the mRNA gets made into proteins. Okay? If you have double the amount of DNA, that's double the amount of spots that are open, so it will produce double the amount of proteins that it should. Okay? This cell will not be normal. It's very important that cells that are in G2 phase don't stay in G2 phase, because they're going to be producing double the amount of stuff that they should, and no cell can sustain that, right? They shouldn't have that much DNA. There shouldn't be that much DNA available to them. Okay? So it's very important that now, once it's in G2 phase, right, that it moves on. And it's going to move on to prophase. And what happens in prophase is that all this chromatin is going to condense. Okay? One of the strands is going to condense into one side of this thing that we refer to as a chromosome. Okay, that's one strand. And the other strand is going to make the other side of this thing that we refer to as a chromosome. Okay, this is a chromosome. Chromosomes have a couple of parts to them. They've got a part that connects them in the middle. This part here is called a centromere. Okay, they've got little portions next to the centromere here where spindle microtubules can attach. These two portions here are called kinetochores. Okay. And then, interestingly enough, there is another way that we can refer to both of these chromosomes, or both of these individual parts of the chromosome, right? So together, right, they make up one single chromosome. But individually, they make up something that are called sister chromatids. Okay, so lots of words that sound similar here: centromere, chromosome, sister chromatids, right? Lots of things that chromatin, right? Those are very similar words, but very important to be able to distinguish between the two. Okay, now when this thing goes through the rest of mitosis, one of the things that happens is that it's going to get yanked apart. Okay, and it's going to get yanked apart now into the two sides. Okay, the sister chromatids are going to be separated from one another. And they've still got all the parts that we would expect them to have. They still have a kinetochore on them. They still have the remnants of the centromere. Okay, but here's the weird thing. Okay, each of these now is a chromosome. Okay, so this is 2 chromosomes. So when they are connected at the centromere, they are a single chromosome, but once they get pulled apart, they're now 2 separate chromosomes. Okay, and we said that in G2 phase, we made a whole bunch more DNA. We made 200% of the DNA that we needed, and that one of the strands of DNA was right here, and one strand of DNA is right here. So now these chromosomes have 100% of the DNA that they need, right? Each one of them is a full copy of the DNA, right? This process where they get pulled apart, that's called anaphase. Make sense? Okay, let's talk about mitosis and the stages of it. So the first one that we're going to draw is not mitosis. Interphase. Not mitosis. Okay? And in interphase, in a eukaryotic cell, what you would expect to see is a cell. The cell happens to be circular, right? And then it's got a nucleus in it, a nuclear envelope, and then inside the nuclear envelope, there's going to be a bunch of chromatin. Okay? The next phase is called prophase. And prophase will look like this. All right, so let's talk about all the things that happen between interphase and prophase. First thing that happens, the nuclear envelope disappears or breaks down. That's one of the things that happens in the transition between interphase and prophase. The chromatin, the all sort of tangled up part, gets all bound together, organized in these things called chromosomes. So chromatin is formed into chromosomes. And the last thing that happens is over here on the corners, you can see these two things have started to appear. These things are called the mitotic spindle, or sometimes they're called the spindle apparatus, and they're mostly made out of microtubules. It's part of the cytoskeleton, right? So we can say that the mitotic spindle begins to form. Okay, and we're going to kind of like snake down here. So here's our next transition. Our next stage is called metaphase. And we say that a cell has entered metaphase when all of the chromosomes are lined up on this imaginary, like, equatorial line that goes across the middle of the cell that is called the metaphase plate. Now, the metaphase plate is not a real thing. It's just like the equator is not a real line, right? If you were like in the ocean or something like that and you crossed over the equator, you wouldn't be like, ah, there it is. It's the line that's on every globe, right? The metaphase plate is also not a real thing. It's just this imaginary line that we say is in the middle of each cell where the chromosomes are going to line up, okay? We had 3 chromosomes in this particular cell. They line up like this, okay? And the way that they line up is that each of the mitotic spindles is going to attach to the kinetochore of these chromosomes here, okay? And they're going to push them and pull them into the correct place, okay? By elongating or shortening, okay? But one of the things that is often skipped over about this is that not all of the mitotic spindles are going to attach to chromosomes. Okay, some of the mitotic spindles are actually going to attach to each other, and this turns out to be really important. Okay, because they are going to be able to now push and pull. They're going to pull on the chromosomes. And they're also going to be able to push on each other. Okay, so we can say that the things that happen between prophase and metaphase are the spindle fibers attached to the kinetochores. And the spindle fibers attached to themselves. And then they push these chromosomes into place. Okay. Now, this is something that is called metaphase checkpoint. Okay, metaphase checkpoint or M checkpoint is extremely important. It makes sure that all of the chromosomes are in the correct place. It makes sure that each of them has a mitotic spindle fiber attached to the kinetochore, and so that's going to ensure that the rest of metaphase is going to go well. It's going to actually work. Okay. A lot of the chemotherapy drugs that we use, the most common type of chemotherapy drug that we use is one that paralyzes the mitotic spindle, and it causes all cells that are dividing at the current time to fail their metaphase checkpoint and then go into apoptosis. Okay? This is important because we said that cancer cells often don't really care about their G1 and their G2 checkpoint, but they still honor their metaphase checkpoint. Okay? So, if a cancerous cell is dividing more often than the rest of the cells in your body, and you have this drug going through your body that prevents the mitotic spindle from moving the chromosomes to the correct spot, moving the chromosomes to the metaphase check or to the metaphase plate, then it's going to fail M checkpoint, and then it's going to go into apoptosis. Now, the side effects of chemotherapy are things like hair loss, weakening of the nail beds, upset stomach, and the reason for that is that those are the other cells in your body that are also dividing very, very rapidly, right? Your nails, your hair, your lining of your stomach are the most rapidly dividing cells in your body, and therefore those cells are also going to be affected because it's very likely that they are also going through mitosis at that given time, and it's affecting all cells that are going through mitosis, not just the cancerous cells, right? You don't hear very much about like muscle cells being affected by chemotherapy, and that's because muscle cells are not often dividing, right? And therefore, they're unlikely to be affected by this paralyzing of the metaphase plate. Yeah.
Well, what if you have like a cut? Do you just like not heal?
Yep. They very much discourage you from getting a chemotherapy treatment if you have an active wound, a wound where you're healing. Well, sometimes depends on the cancer. You're like, okay, is it going to be better for you to heal from this, or can we just throw you on some antibiotics and get you your chemotherapy that you need? That's just how severe the cancer is. Yeah.
You said yesterday that your liver is constantly making cells, right?
Yep, liver's pretty affected by it too.
Okay.
Yep. All right, so we move on. We go from metaphase. In the book, it talked about something called prometaphase, right? Prometaphase is this weird sort of time in between prophase and metaphase. It's not technically in metaphase until all of the chromosomes are on the metaphase plate, right? So let's say 2 of the chromosomes are on the metaphase plate, and one of them was still chilling out up here, right? It's not in prophase anymore because it doesn't look like this. It started to connect, but it's not fully in metaphase anymore or yet because it's not all lined up on the plate. So if it's not quite there yet, you would call that prometaphase, right? Okay. So the next one. It's called anaphase. Why is it shaped like that? Okay, it's shaped like that because this is what happens in anaphase. The cell begins to elongate, right? And it is elongating because these spindle fibers here are pushing on each other, right? So these spindle fibers that are attached to the chromosomes, they want to pull to get the chromosomes to come back, but the other ones are actually going to push on each other, and that's going to cause the cell to elongate, right? So you've got Chromosomes have now been pulled apart here by the mitotic spindle, but you've still got mitotic spindle fibers that are attached to each other that are pushing the cell to cause it to elongate. So spindle fibers attached to kinetochores. Pull sister chromatids apart. And spindle fibers attached to spindle fibers push to elongate the cell. Okay. That moves us into telophase. Which I always learned as telophase, but if you watch videos on the internet, it's like people call it telophase, like telo, like 90% of the time. I've never, right? All my teachers call it telophase. I don't know if it's like an American thing versus a British thing or something, but everybody's always calling it telophase. So if you hear that, that's what it means. Okay. So we're still elongated at this point. Oopsies. But we are now starting to see the beginnings of cytokinesis happening. Okay, you got these little pinches on the side, and the reason for this is that there is an actin filament, contractile filament in there that is pulling the sides of the cell wall together, or the cell membrane together, rather. Okay. Telophase, other than that, is the exact opposite of prophase. Okay, so you start to see the nuclear envelope reappear. Okay, but it reappears now on both sides. Those chromosomes that we had before, they break back down into chromatin. You might see the spindle a little bit, but for the most part, the spindle is all broken down. So lots of important stuff happens here, right? We can say that chromosomes break down into chromatin. We can say that the spindle breaks down. And the nuclear envelope reforms. And generally, near the mid portion of telophase, when the nuclear envelope is just starting to reappear, that's when we start to see cytokinesis beginning. Okay, so there is not going to be a huge defining line between a cell that is going through cytokinesis and a cell that is in telophase. Right? They're going to look very similar to one another. The only thing that I want you to know about cytokinesis is that there are different types of cytokinesis. Okay, based on whether it is a cell with a cell wall or a cell without a cell wall. So an animal cell or a plant cell. Okay. So the first type that we're going to look at here is an animal cell. And in animal cells, they're going to look something like this. Okay, and the middle part here, that's where you've got your contractile filament. Contractile actin filament. And it's pinching the cell together, and that's called, that part where it's being pinched is called the cleavage furrow. Okay, and that is in contrast to plant cells. Plant cells don't have that. Plant cells are more square, rectangle-y. Right? So here's a plant cell, I guess. Sure. And you're going to be able to see the nucleus in here still. And here's our chromatin. But the way that this works is that a cell wall begins to form in the middle, called the cell plate. And that cell plate then just grows to either side, and as it grows to either side, it then will separate the two cells, and so each one will then be half the size of the previous cell. Okay? How does a plant cell grow then? How does it get bigger? What? How does it stretch out? What does it use? Guys remember? Use the central vacuole, right? So it fills up its central vacuole with water, and because there's this box around it, it stretches out that box that's the cell wall, because it's got to fill up that box and stretch itself out, right? So, I wonder if I can make this work. This could go poorly. Computer is getting old, so I'm not sure it's going to like pulling up a camera when I have my recording program up. But we'll see. We'll give it a go. So Thursday, we are going to do the microscope lab for looking at mitosis. Yeah. But I wanted to show you something interesting about that. First today. What did I do with my pen already? Okay. Alright, let's see how much my computer hates this. It's gonna do it. Alright. So here we go. This is an interesting thing because this is the root of an onion plant, and it has been dyed to the point where only the DNA is showing up. So there's a lot of other stuff in these cells, but only the DNA is blue. Okay, and so really it's all we're going to be looking at. Okay? Now, the interesting part about that is because the way plants grow, they grow only at what's called their meristem. And so the only points where you're going to see any cell division happening are at the very tip of this plant, because the rest of the plant, it's already grown, and so it's these cells are all in interphase. And if you zoom in on them, you can see that they're in interphase. Maybe. You can see that they're in interphase because you can't make out any chromosomes in here, right? You can't definitively see any chromosomes in those. But if you travel down a little bit to like here, you can start to see some cells where there's some chromosomes, right? You guys see the chromosomes in here? Where does it look like they're lined up? On that metaphase plate. And that's different than this one that just has just a whole bunch of chromatin, right? There's another one with chromosomes in it over there. Trying to figure out where you guys are looking compared to where I'm looking. All right, so you can see this one over here. The chromosomes are not really doing anything. So this is probably like in early prophase here. This one's in like that weird pro-metaphase state because here's the metaphase plate, and then here's this one chromosome that's just like hanging out. It's not quite there yet, right? So the spindle's trying to move that one around. Yeah, so this is what you're going to be doing on Thursday. We're not going to be here tomorrow, right? Because it's PSAT day. So yeah, you don't need to do anything special. You don't need like closed-toe shoes or anything for this lab. It's just a microscope lab. But we'll be looking at this stuff, which is onion root tip, and we'll also be looking at fish embryos, fish blastulas. What's up?
So is that part like when you zoom down, like at the very bottom? Is that like the bottom of the root, or is that like the top?
It's the bottom of the root. Yep. And so another interesting thing about that, right, is there's a whole bunch of cells out here that are dead, right? So these cells are part of something called the root cap, and the reason why the root cap is there is because this is the like most important part. This is where all the new cells are, right? You can see how dense everything is here, how much smaller the cells are, and so this part is pushing through the ground, right? If you imagine like sandy soil pushing through the ground there, this outer part gets destroyed like non-stop because it's just like rubbing itself against sandpaper. Right? And so all these cells are all just dead cells that protect these middle parts here, which is called the root apical meristem.
So are those cells like actively performing mitosis, or is it?
No, they are dried. So we cut them and then stain them, yeah, and then they're dried. So they're stuck. They're stuck in whatever point they were. Okay? Questions? Yeah.
What happens to all the organelles when it's